Hello, everyone, and welcome to today's webinar. My name is Catherine Heldy, and I'm the Chief Scientific Officer here at Syngap Research Fund. Our presentation today is Targeting Gamma Oscillations to Improve Cognition. I have the pleasure to introduce today's speaker, Dr. Vikas Sohal from the University of California, San Francisco. We met Dr. Sohal when he applied for a grant from SRF, and Mike had the pleasure of visiting his lab. We are so excited to be funding project that is outside the realm of other Syngap research that we have seen, and he's going to tell us about that today. Dr. Sohal studied applied mathematics at Harvard and Cambridge before completing his MD and PhD degrees at Stanford. During graduate school, he studied alma-cortical oscillations. He stayed at Stanford for a psychiatry residency, and during that time, he carried out some of the first experiments using optogenics, including closed-looped approaches to study the mechanisms and functions of gamma oscillations. In 2010, he started his independent laboratory at UCSF, which uses many techniques: optogenics, electrophysiology, calcium imaging, genetically encoded voltage indicators, and computational modeling. Study how emergent patterns of cortical circuit activity contribute to cognition and emotion. This has led to several discoveries with important implications for neuropsychiatric conditions, including schizophrenia, autism, anxiety, and depression. Dr. Sohal has received a whole slew of impressive awards for his research, and he is a board-certified psychiatrist and continues to see outpatients in the early psychosis clinic at UCSF. A recorded version of this webinar will be available on the SRF website under Webinars, located on the News and Events menu. By the end of this presentation, you will have the opportunity to get answers to your questions. We'd love to hear from you. Please write questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker today is Dr. Vikas Sohal, and his presentation is Targeting Gamma Oscillations to Improve Cognition. It is now my pleasure to turn things over to Dr. Sohal.
All right, thank you so much for that introduction. And I just want to say, we're so fortunate, obviously, for the, the support for our research and the opportunity to tell you about it, and the financial part of it, but also just from getting turned on to this area of obviously very important research is really something that SRF kind of initiated. I got an email from Mike several years ago, and he pointed out that probably what we were doing could be very, very relevant, to thinking about Syngap and that there could be a lot of synergy here. And I think that's turned out to be totally true. And this is going to be a wonderful and exciting and hopefully in the long term impactful research direction for my lab. So I want to thank SRF for not just the financial support that we're just getting started with this project, but honestly for the intellectual interaction, which I hope will continue with this talk today. All right, so let me jump into it. I am going to talk about a few different topics here. You'll see there's a very strongly connected train of thought. I'm going to introduce gamma oscillations and how I originally became interested in thinking about them in the context of schizophrenia. And then I'm going to talk about 3 published studies from my lab, which all build on each other, really demonstrating this idea that gamma oscillations could be important for cognitive function. And then I'm going to tell you about the preliminary results that led to this funded application from SRF, thinking about how these are relevant to Syngap. In this case, Syngap heterozygous mutant mice, but hopefully to treating conditions which involve Syngap. All right. So most of the work I'm going to talk about, these 3 projects, were all led by an amazing former postdoc, Kathleen Cho, who now has her own lab, her own group in Paris. And then this work on Syngap was done by a current postdoc in my lab, Shiyu Zhu, who is really very, very passionate about thinking about neuropsychiatric conditions and about development. And so this has been a perfect fit. And I think the work he's doing is super exciting, as you'll hear. All right, so let me start with background. So I'm a psychiatrist, and I mostly work with patients with schizophrenia. I just want to make sure, like, if my messages and stuff pop up, are they showing up for you guys or no? Okay, I'm going to take that as a no.
Nope,
No,
we're
you're
good.
good.
Yeah, that's why I wanted to make sure. A couple had popped up, and I was like, I don't think they're showing up. Okay, so you know, and when you hear the word schizophrenia, right, mostly we think about paranoid delusions and hearing voices, and those are symptoms which are indeed very distressing for patients and their caregivers. But honestly, they're not the things which end up making the biggest difference in terms of how people are able to get on with their lives. Are they able to live independently, work, study, maintain social relationships? Actually, there are cognitive deficits which tend to precede and persist, and outlast the other symptoms. And these deficits are in what we often think about as executive functions, higher order aspects of cognition, mediated by parts of the brain like the prefrontal cortex. And those are the best predictors of whether or not people can, really live their lives to the fullest. And an example of one of these cognitive abilities is this task that we call the Wisconsin card sorting task. Some of you might be familiar with this. I'll show you kind of how it works here. So in this task, you'd be given a card with, say, 2 red crosses on it and told to sort that into one of 4 piles. And you wouldn't be told the rule to use. So you could sort based off of color or shape or the number of objects. You would be told if you did this right or wrong. And pretty quickly, you would infer what the rule is. Okay. And then without warning, the rule would change. So instead of sorting based off of color, you would need to realize that the rule has now changed to a shape-based rule. Start ignoring color and start sorting based off of shape instead. Okay. And so we want to study this kind of ability in mice, and we can't create a perfect analog of the Wisconsin card sorting task, but we want a task which requires, individual subjects to learn a rule, realize when that rule has changed, and then when it changes, kind of suppress their old behavior and start sorting based off of a new strategy, okay? And so we use this task in which mice dig in bowls to find a hidden food reward. And in this task, each bowl will have a different odor and texture cue. So one might smell like garlic and be filled with sand, and the other might smell like coriander and be filled with like wood chip or else like fine litter or something like that. And the mouse will, perform this task, and it'll pretty quickly realize that one of these 4 cues, in this case, the smell of garlic, indicates the location of the food reward. And when the mouse learns that, then without warning, the rule changes from an odor-based one to a texture-based one, or obviously vice versa. So this is what we call a rule shift, okay? And it encompasses these same aspects of having to realize that the rule has changed and then adapt your behavior and suppress your old behavior. Okay, so what sorts of things in the brain do we think are important for this ability? Well, one of the most consistent findings in schizophrenia is that there's a particular class of neurons. These are called parvalbumin interneurons, or sometimes abbreviated as PV interneurons. And these seem to be abnormal, particularly in the prefrontal cortex. So this is a post-mortem sample of brain tissue from healthy control or individual with schizophrenia. This is looking at levels of the enzyme which synthesize GABA, the inhibitory neurotransmitter that these neurons use to communicate with other neurons. And you can see that levels of this enzyme are lower in schizophrenia, and they seem to be particularly lower in parvalbumin expressing interneurons. And actually levels of parvalbumin, the marker for this class of cells, are also decreased. Okay, so there seems to be something wrong with these parvalbumin interneurons, which raises the question of what they normally do in the brain. And there's a lot of things that they do. I studied these when I was a postdoctoral fellow with Carl Dijsselroth and a psychiatry resident. And one of the things that they do is they generate what are called gamma oscillations. Now, many of you have probably heard of brain waves, things like alpha, beta, gamma waves. And the reason is because these are patterns of brain activity that you can see if you put like EEG electrodes on the scalp, the kind that are used to identify seizure activity. You'll constantly see these rhythmic up and down fluctuations in brain activity. And these fast fluctuations, which occur something like 30 to 100 times per second, these are called gamma oscillations or gamma waves. And this is an example in a cat. When a visual stimulus, a moving bar, is shown, you see the appearance of these gamma waves. And then if you're recording from a single neuron, you'll see that neuron tends to spike. And in this example, you see that these are the troughs and the peaks of the gamma wave. And you see that the spiking of this individual neuron is aligned to this gamma oscillation. So when we talk about brain waves or brain oscillations, this is what we're talking about. We're talking about a pattern of activity that rhythmically fluctuates. You can see it at a lot of different levels: scalp EEG, electrodes inside the brain, and even the spiking of individual neurons. And that's what a gamma oscillation is. Now, gamma oscillations happen in all different parts of the brain. And if they're generated by parvalbumin PV interneurons, you might think that in conditions where those interneurons are not working quite right, as seems to be the case in schizophrenia, something would happen to those gamma oscillations. And so many people have studied gamma oscillations in individuals with schizophrenia as they're performing the sorts of cognitive tasks that might be challenging for them. This is an example of one of those tasks, right? You would be shown a green or a red square. The green square indicates the easy condition, where when an arrow pops up, you move your hand in the same direction as the arrow, so you move your right hand. But when a red square pops up, it's the hard condition. And so even though the arrow is pointing to the right, you have to respond with your left hand. This is a kind of task which, again, uses this kind of like cognitive control. You have to suppress your tendency to do one thing and use a different strategy to do a different thing. And so people record EEG during this task. And in healthy subjects, if you measure, essentially this is the size of wiggles at different frequencies. So the size of these rhythmic fluctuations at 30, 50, 80 cycles per second, you can see a lot of red and orange here, which means there's a lot of these gamma oscillations that are being recruited by this task. And in individuals with schizophrenia, these gamma oscillations are deficient, okay? And so kind of based off of what I've told you so far, many people, this was originally really led by David Lewis, a psychiatrist at the University of Pittsburgh, came up with this idea that when these parvalbumin interneurons that I told you about, when they're dysfunctional, that might cause deficits in these task-evoked gamma oscillations. And maybe these oscillations are important for how the brain works. So when they're deficient, that causes cognitive deficits, like difficulty switching between different rules. Okay, so this is this basic idea that gamma oscillations are important for cognitive function and maybe really important in the context of conditions such as schizophrenia. But there's a problem. And the problem is that for decades, whether or not gamma oscillations are actually important has been a hugely controversial subject in neuroscience. And there's a few reasons for that. And again, this is maybe not so critical for non-scientists out there, but some of you might find this interesting. Okay, so the first is that, people see these oscillations and they come up with ideas about what they're doing, like, oh, maybe when these oscillations are synchronized, that means when the peaks and troughs in two different regions line up, that helps those two regions communicate and exchange information. Okay, that's a great idea. It's a very common idea. It makes a lot of sense, but there's been no direct evidence for that. And there's lots of ideas about how gamma oscillations might work, but very little direct evidence that they're actually doing something meaningful. Okay. The second is that anytime you have neurons in a circuit and they're connected up to each other, they tend to generate oscillations, okay? It's just sort of like anytime one group of neurons gets excited, it's going to recruit other neurons, which are going to inhibit those, and then that inhibition will take time to wear off, and that leads to rhythmic increases and decreases in levels of brain activity. Okay, so many people say, like, look, these oscillations, they just happen. They tell you the brain is active. They tell you cells are connected to each other and talking, but they don't actually do anything. And the third is that, just like your car radio can pick up a radio station that's the same one as a radio station that another car radio will pick up, it doesn't mean that the radios in those cars are like there's a person talking there that's producing the voice or the music. There's a signal coming from elsewhere, and the cars are just picking up that signal. And the same way, when we have electrodes on the scalp or in the brain and we pick up oscillations and we see that they're synchronized, it doesn't mean that the underlying brain regions are synchronized. There could be a signal that's propagating through the brain the same way that radio waves propagate through the air that's being picked up at both those locations, okay? And so this is an important controversy because in the context of schizophrenia, we find these problems with gamma oscillations, but we don't know, are these just biomarkers that tell us that the circuitry is abnormal? Like these are just reflections of what's going on and telling us that, hey, something's not quite right? Or is this like the way the brain works? And so when these oscillations are not synchronized, it tells us that that's an important thing that we have to correct in order to restore cognition. Okay, and so this is the basic question that my lab has been really focused on for the last 10 or 15 years. All right, and so I'm going to tell you about now quickly these 3 studies where I think we've made a lot of progress on this question, and then finally I'll tell you how this is relevant again to Syngap. Okay, so we started studying these mice that have mutations in 2 genes, DLX5 and DLX6. I'm sure you guys, families are used to hearing about lots of genes. And DLX5 and 6 are genes that are particularly important in the cortex of the brain for the development of inhibitory neurons like parvalbumin interneurons. And they've been widely studied by our collaborator, John Rubinstein, who's a child psychiatrist here, who's kind of an expert on neurodevelopment. And he suggested that we study mice that lack 1 copy of DLX5 and DLX6 because he thought they might have something wrong with their parvalbumin interneurons that might tell us something about what happens when these interneurons are not working quite right. And so we studied these mice, and quite interestingly, they were initially normal when we studied them like during puberty and at young ages. But when we studied them sort of at young adulthood, we saw that the parvalbumin interneurons became abnormal. And so in this case, we record electrical activity from those parvalbumin interneurons in wild type or mutant mice. And as you can see here, these parvalbumin interneurons have a very characteristic firing pattern. They can fire at very high rates. And these action potentials maintain their shape, and they're very, very narrow. But in the mutant mice, we saw that these action potentials seem to kind of run down over time and become sort of shorter and squatter. And this is important because the ability of these neurons to fire very consistently at high rates is thought to be important for their ability to participate in fast brain activity like gamma oscillations. Okay, if we then had these mice perform our rule shifting task and measure how many trials it takes them to learn an initial rule or a rule shift, we saw that the mutants shown here in blue do just fine on the initial rule, but they have a lot of trouble with the rule shift. And the reason is because they get stuck on the initial rule. They're perseverative and can't update to follow the new rule. And then finally, we implanted electrodes in the brain and measured kind of the change in activity at different frequencies. So slow waves, fast waves. And here's just a bar plot which summarizes what we found. In black is what happens during the rule shift in normal mice. And then in blue is what happens in these DLX5/6 mutant mice. And we see that in this rule shifting task, there's a big increase in activity in this broad gamma frequency range, but that activity is blunted or deficient in these DLX5/6 mutant mice. So they seem to recapitulate some of these things: abnormal parvalbumin inhibitory neurons, problems with this rule shifting task, and problems with gamma oscillations that are seen in schizophrenia. And these seem to be tied to abnormal development, maybe, of these parvalbumin interneurons. Okay, so at this point, we thought about like how to try to fix this. And I mean, I know there's a lot of scientists on here, and many of you are probably familiar with optogenetics. But for those of you who aren't, I'll talk about this for a second. So optogenetics is a technology that was really pioneered in the lab where I was a postdoc and psychiatry resident, the lab of Carl Deiseroth in the mid to late 2000s. And I actually used to see patients with Carl. He's also a psychiatrist. And we saw one patient, a young man with, probably on the autism spectrum with ADHD. And he actually worked as an engineer, and he came in to see us one day and he said, "Oh, Dr. Deiseroth, I've been telling my friends about your research and showing them YouTube videos and explaining to them how you take these genes from like algae and put them in the brains of mice and then use lasers to control their thoughts." And that might sound like some kind of delusion, but it's actually a perfect description of how optogenetics works. The idea is that single-celled organisms, algae like Chlamydomonas or Neutronomonas, they have these single proteins that are sensitive to light. And when they're activated by light, they either allow electrical signals via ions to flow into cells, or they might actually pump ions into cells, and this can either excite or inhibit the activity of a neuron. And so we can use the modern tools of genetic engineering to put these ion channels, which are activated by light, into specific neurons in the brains of mice, and then we can deliver light in order to activate or suppress the activity of those neurons at desired times, right? So we can deliver flashes of blue light to stimulate neurons with channel rhodopsin, or we can deliver continuous like orange light to inhibit the activity of neurons with halorhodopsin. Okay, so we thought, hey, if problems with gamma oscillations are really what are causing these cognitive deficits in these DLX56 mutant mice, maybe we can use optogenetics to boost their gamma oscillations and try to fix these cognitive deficits. And so we did an experiment where we took DLX56 mutant mice, we injected a virus to express channel rhodopsin in inhibitory neurons and specifically parvalbumin interneurons in the prefrontal cortex of these mice. We implant these little fibers so we can then deliver flashes of light, and we can flash light at 40 hertz, 40 times per second, to try to boost gamma activity. And we do that basically on the second day of testing after mice have learned an initial rule and when they're starting to do a rule shift, okay? And so here I'll show the performance of two groups of mutant mice. One group is a control group that does not have channel rhodopsin, shown in white, and then in yellow is a group which does have channel rhodopsin in their inhibitory neurons. And basically, this is how long it takes them to learn the rule shift. And you can see on the first day, they all take a long time to learn. Okay, they're all very perseverative and stuck on the initial rule. But on the second day, the group that's getting this kind of boost to their gamma oscillations via these 40 hertz light flashes that should activate channel rhodopsin, the group that has the channel rhodopsin in their parvalbumin neurons suddenly starts to perform much better. In fact, they perform just like normal mice. And what was really surprising to us is that when we then tested these mice the next day, we didn't deliver any more light flashes. They continued to perform normally. In fact, we could test them a week later. We've now done this like basically a month later, and they will still perform more or less normally, the group that got light flashes one time. Okay. And so this was a super exciting result because we sort of cured, this cognitive problem via one session of appropriately targeted stimulation, okay, in these mice. And, this was honestly something that was way better than we expected. And it was very intriguing and very exciting. That said, we've obviously continued to study this because we didn't really understand what was happening, okay? We didn't know if we got this pro-cognitive effect because these 40 hertz light flashes are just like, this is the right way to kind of like tickle these neurons to make them respond, or is it because actually the gamma oscillations are really important, right? So we didn't know if this worked because we were just stimulating these neurons at a frequency that they like, or because gamma oscillations are really important for how the brain works. And so that was a question we had. And we also, I think the focus of this is on, therapeutics, but we also are, I'm a neuroscientist and I didn't know like, okay, we found this really important role for gamma stimulation in mutant mice, but does that tell us that this is important normally? Like what's going on here? Okay, so that's really the second story that I'm gonna briefly tell you about now. And one thing about this is we decided to look in normal mice for when gamma oscillations are recruited. And for this, we decided to use not electrical recording of gamma oscillations, but these genetically encoded voltage sensors. So what I'm talking about here are proteins, right, which are fluorescent proteins shown here in green, which change their fluorescence based on the voltage, based on the activity of the cell they're in. Okay, so when this cell becomes active, we could detect that via change in their fluorescence. Now, this is optical instead of electrical-based way of measuring neural activity. And the reason that's important is because now when we measure these light signals from, say, parvalbumin interneurons in the left and parvalbumin interneurons in the right prefrontal cortex, we can see whether those signals are synchronized. And if they're synchronized, we're not worried about what we're worried about with electrical signals because we know that the light is coming from the neurons right under our optical fiber. Okay, so we know that means that actual neurons are synchronized, and we know it's actually these neurons which we are causing to express this fluorescent protein. I won't go into this in detail. We also have in red a reference sensor that allows us to control for changes in like the fluorescence that we detect that's just due to like the optical fiber bending or even like blood flow and things like that. And so we collect all of these signals from the left and the right sides of the brain so we can look at how synchronized parvalbumin interneurons are on both sides. We kind of filter the signals in a frequency band of interest, in this case around 40 cycles per second, to focus on gamma frequency activity. And again, we have a mathematical procedure for kind of calculating how synchronized these signals are that I could describe more if people are interested in. It's described in this paper. Okay, so basically the big finding is that when we do all of this work to measure how synchronized is rhythmic activity between parvalbumin interneurons in the left and the right prefrontal cortex, what we find is that synchrony goes up after error trials during the rule shift. Now, this is super interesting because error trials are very important, right? We don't tell mice, hey, the rule is changing, you got to change your behavior. What happens is the mouse learns one rule, it's following that rule, and all of a sudden, it doesn't get the food reward it's expecting, okay? And that lack of a reward, that incorrect outcome, should tell the mouse like, hey, this rule you previously learned, it's not working anymore, and you got to update your strategy. And at that moment, we see this increase in the synchronization. That means the lining up of peaks and troughs of these gamma rhythms between parvalbumin interneurons in the left and the right sides of the brain, specifically in the prefrontal cortex. Okay, and we don't see this increase after correct trials, and we don't see it in other frequency bands. We only see it around 40 cycles per second. Okay, so this tells us that, yeah, these gamma oscillations are happening in the brains of normal mice. They're happening at the right time to be important. Are they actually important for normal mice to learn? And how can we think about this fact that we see synchronization between the two sides of the brain? Okay, so what we realized is that we, like many people, deliver optogenic stimulation. I told you in the previous part about how we could deliver rhythmic stimulation to boost rhythmic activity in order to rescue activity in DLX56 mice. Now, whenever we do this, we always deliver stimulation to both the left and the right side of the brain. And normally we just deliver the same exact light flashes. But what we realize is we don't have to do that. We could deliver 40 hertz, so rhythmic patterns of light flashes to the left and the right side of the brain. They could be perfectly aligned, in which case they not only boost rhythmic activity, but they also synchronize or line up that activity across the two sides of the brain. But we could also deliver rhythmic activity that's misaligned. So 40 hertz activity to the left side, 40 hertz activity to the right side, but the peaks and troughs are misaligned. So it shouldn't actually increase synchrony of that activity across the brain, okay? In fact, it should disrupt the normal pattern of synchrony. And when we do this in previously normal mice, so these are mice that now have channel rhodopsin, so in their parvalbumin interneurons, so we're boosting gamma activity, but we're misaligning it across the two hemispheres, that actually makes normal mice very perseverative and stuck, just like the DLX56 mutant mice previously were. We can try different patterns. So we can try misaligned 20 hertz activity, so outside of the gamma range, misaligned 40 hertz activity, and then aligned 40 hertz activity, the same pattern we used previously in the DLX56 mice. And only this misaligned 40 hertz activity ends up causing perseveration, causes mice to get stuck on the old rule and have trouble learning a new rule. Okay, this made us think we should now go back and see what's happening in our DLX56 mutant mice. We should measure gamma synchrony the same way I told you about before, and we should look at the difference between error and correct trials, because I previously told you that in normal mice, which are shown here in gray, there's this increase in gamma synchrony on error trials compared to correct trials, specifically around 40 cycles per second, so in this 30 to 50 hertz range. Okay, so here we see this evidence of increased gamma synchrony in the wild type or normal litter mates of our DLX56 mutants. But when we look at the mutant mice, we no longer see this normal increase in synchrony, okay? And then we can try these different patterns of stimulation. And I'll note that both on day 2 and day 3, we're boosting gamma activity, but one day we're doing it in a misaligned way, and the other time we're doing it in an aligned way across the two sides of the brain. And only when we have this proper alignment between gamma activity on the two sides of the brain do we see an improvement in the performance of these mice, delivering almost the same pattern of activity, but just having it kind of be jittered between the two sides of the brain doesn't improve the performance of these mice, and they still are perseverative and take a long time to learn. Okay, now for all the aficionados out there, I'm just going to mention we did all these same experiments, but we targeted a different group of inhibitory neurons, some myostatin expressing inhibitory neurons, which is another big group. And we don't see any of these things. These neurons don't seem to synchronize, and stimulating them doesn't cause problems when we do it in a misaligned way, and stimulating them in DLX56 mutants doesn't help if we deliver synchronized stimulation. The other kind of interesting thing is everything I told you only seems to be important for this like rule shift where we go from an odor-based rule to a texture-based rule. If we look at learning of an initial rule, or we look at switching from like garlic to coriander, so like a like within odor change in the rule, actually gamma synchrony doesn't seem to be important for any of that. Okay. All right. So what I've told you so far is that it's not just the oscillations, but actually them being aligned and synchronized, in this case, across the two hemispheres, that seems to be really important for rule shifts. And we found, what for scientists is, we call it a double dissociation. It's a super cool effect where we took two patterns of stimulation that are almost exactly the same. One of them is slightly misaligned, and the other one is perfectly aligned, and they lead to two completely different effects. So the misaligned stimulation actually causes perseveration in previously normal mice, but the perfectly aligned stimulation in mutant mice can actually normalize and their performance and eliminate their perseveration. Okay, and so for me, this was super direct evidence that this concept of oscillations being synchronized is actually really important for brain function, okay? Because the only thing that differs in these two cases is how synchronized or how well aligned the rhythmic activity is on the two sides of the brain, and that completely changes the effect of the stimulation. Okay, now that said, we still don't know where exactly the synchrony comes from and what it does. And I'm just going to very briefly tell you about a third study where we started to get at these questions. But before I do that, I'm going to tell you about a tangent, which is actually really interesting when you think about therapies and how these kinds of findings might be translated to the clinic. Okay, so it turns out that we don't have to use all this fancy technology that neuroscientists love to use in order to increase the synchronization of gamma activity in the brain. We could actually use a really old drug, clonazepam, which is sometimes called Klonopin, which is a benzodiazepine, which I'm sure both obviously clinicians, but also a lot of families might be familiar with. It's sometimes used, for seizures, for sleep, for anxiety. We tried using sub-anxiolytic and sub-sedative doses of clonazepam. And the reason we did that is because this targets the GABA receptors that the parvalbumin interneurons are normally using to communicate with other neurons. And so this might be a way to kind of boost the output from those parvalbumin interneurons in order to try to rescue it in disease states. And we use really low doses because we don't want to cause sedation that might interfere with cognition. Okay. So we can take our DLX56 mutants, we can test them with vehicle injections or clonazepam injections. And what we find is that the clonazepam also improves the performance of these mice. It gets rid of these perseverative errors. It makes them learn much faster. And if we look at gamma synchrony, it seems like actually gamma synchrony goes up specifically after incorrect choices when we give the mice clonazepam. And interestingly, only gamma synchrony goes up. We don't see increases in other frequency bands, only around 40 hertz. And we don't see increases in synchrony after correct trials or at baseline. So this is just kind of a curious and very intriguing finding. It's kind of like when you hurt one part of your body, you get a sprained ankle, and you take ibuprofen, and your kid might be like, how does the ibuprofen know to only go to your ankle? It doesn't, but somehow it only makes your ankle feel different. The same way here, the clonazepam, even though we're like giving the mice these drugs that should act all over their brain, it only seems to increase synchrony exactly at the right time. So that's kind of an interesting kind of finding that I can talk about more if people are interested. Okay. All right. So now let me tell you where this synchrony comes from and some ideas about what it does. Okay, my lab, obviously, we study other things. And one of the things we've been studying are long-range inhibitory connections in the brain. So there's this idea that in the cerebral cortex, inhibitory neurons mostly send connections locally, just to their neighbors. And that the only long-range connections are basically excitatory connections. But we had previously found that this isn't true in the prefrontal cortex. We were inspired by some other people who were thinking about this, and we had found two examples where in the prefrontal cortex, inhibitory neurons like parvalbumin interneurons actually send long-range projections to other parts of the brain. And so if we're thinking about how like the two sides of the brain synchronize, these long-range projections are a natural kind of source of communication that might be important for that. And so we did a very simple experiment where we labeled parvalbumin neurons in one prefrontal cortex with channel rhodopsin, and in this case, a fluorescent protein YFP. And then we look on the other side of the brain, and we see that indeed there are fibers that are crossing the hemispheres, and we call these callosal projections. They go through the corpus callosum, and we see them in the opposite side of the brain. And we can stimulate them and actually record synaptic responses in the other side of the brain. So this is all just basic synaptic biology to show you like, oh, there's actually these long-range inhibitory connections going from parvalbumin interneurons on one side of the brain to the other side of the brain. And actually, the reason parvalbumin interneurons are called interneurons is that it's thought that they only project locally, which is why they're called interneurons. But this is not actually true. Some of these are actually long-range projecting neurons that go to the other side of the brain. So now we thought like, hey, we should target these long-range fibers and see if shutting them off is sufficient to like shut off, rule shift learning. And so we did exactly that. We used that other tool, halorhodopsin, which can be used to inhibit neuronal activity. We put that in parvalbumin neurons on one side of the brain, so it will go into their fibers on the other side of the brain. We then stick a little optical fiber here so we can deliver light and inhibit just the synapses from parvalbumin interneurons on one side of the brain going to the other side of the brain, okay? And when we do that in previously normal mice, okay, the mice that have halorhodopsin, when we deliver light to activate that halorhodopsin, they now take much longer to learn these rule shifts, and they become very, very perseverative, okay? We then do essentially the experiment I told you about before, where we use these genetically encoded voltage indicators to measure synchrony between parvalbumin neurons on both sides of the brain. We now do this while we also have halorhodopsin so that we can inhibit these parvalbumin interneuron connections to the other side of the brain. And in normal mice, again, from even when we deliver light to activate halorhodopsin, these are mice without halorhodopsin, we see this increase in gamma synchrony after error trials. But in the mice which have halorhodopsin in their parvalbumin interneurons, when we deliver light to activate that halorhodopsin, and now we're not shutting off the whole neuron, we're just shutting off its synapses on the other side of the brain, that's enough to get rid of this increase in gamma synchrony that normally happens after errors compared to correct trials during a rule shift. Okay, so we think this is why mice are having trouble learning the rule shift, is we've gotten rid of the gamma synchrony. Okay, so let me just summarize what I've told you. I've told you that we have parvalbumin interneurons in the prefrontal cortex on both sides of the brain. We see that they actually synchronize their rhythmic gamma frequency activity while mice are learning a rule shift, and this is necessary for mice to learn the rule shift. I'm not going to tell you about this in detail, but if we look at patterns of activity, so here this just represents like a pattern of activity across many, many neurons. Patterns of activity in the prefrontal cortex change when mice learn a rule shift, just sort of how we think the brain works. And what we have found now is that these parvalbumin neurons send connections to the other side of the brain. And if we inhibit just these connections, that gets rid of gamma synchrony. It prevents mice from learning a rule shift. They get stuck on the initial rule. And actually, if we look at what happens to patterns of activity, the patterns of activity no longer update. So this gamma synchrony seems to be really important for updating patterns of activity so you can learn new rules. Okay, that's the basic message here. Now, one thing that's going to tie back to what I told you about earlier, remember we saw these long-lasting effects in the DLX56 mutants. We stimulated them one time, and we saw that we kind of cured them. We've always been wondering, like, how does that work? And so one thing that was interesting was we also see long-lasting effects when we target these callosal or cross-hemispheric projections from parvalbumin neurons. So we did this experiment I told you about before. Now we're going to put both halorhodopsin and channel rhodopsin into our parvalbumin interneurons. So we can either inhibit or stimulate them. We have two groups of mice here. One has just halorhodopsin, so we're going to inhibit the parvalbumin interneurons connections. The other one has halorhodopsin and channelrhodopsin, so we can stimulate them. On day one, we deliver light to inhibit these projections, and previously normal mice have trouble learning a rule shift. On day two, we don't deliver any more light, but it turns out the mice still have trouble learning a rule shift. So we can induce a persistent deficit in the performance of these mice. Now, if we stimulate just the long-range projections from one side of the brain to the other, we actually rescue this performance. And that turns out to be a long-lasting effect. So we can stop delivering light and test the mice again. So I told you there's this kind of like magical plasticity where all of a sudden the mice seem to be cured. And we think that maybe these long-range projections going from one side of the brain to the other are important for that plasticity. Okay, so this brings me to what I'll talk about in the last couple minutes, which is how all of this relates to Syngap mice. And as I think is clear, we think this could be a powerful mechanism for kind of improving cognition, targeting these long-range GABAergic synapses that are formed by these parvalbumin interneurons. Okay, and it turns out that parvalbumin interneurons also seem to develop abnormally in Syngap mutant mice. And so we were really inspired by these papers. These were brought to our attention actually by Shilpa Kadam, who had reached out to us and suggested we start working on Syngap. And just to make a long story short, it looks like levels of parvalbumin are lower in Syngap mice. The synaptic output from these parvalbumin neurons is lower in Syngap mutant mice. And actually gamma oscillations are disrupted in these Syngap mice. And this was found in this nice paper from Graziella De Cristo's lab, and similar reductions in parvalbumin neuron activity were also found in Shilpa Kadam's lab. Okay, so we did a very simple experiment. We said, let's test how Syngap mutant mice do compared to their wild type normal litter mates on this kind of like rule shifting task. On day one, we won't do anything. On day two, we'll stimulate the parvalbumin interneurons at 40 hertz and see what happens. And then we'll test them again a couple of times after that. And so this is early days, but this is what we see in Syngap mutant mice. They take a long time to learn the rule shift. They get stuck on the first rule. They're very perseverative, just like the DLX56 mutants. They don't perform like normal mice, but when we deliver this gamma frequency stimulation, all of a sudden the Syngap mutant mice perform normally. And what's, again, to me, just totally remarkable is they continue to perform normally a day later or a week later. This is very, very early, but we've tried stimulating just on one side of the brain, stimulating the cell bodies of neurons, or stimulating the terminals. So these terminals that go from one hemisphere to the other hemisphere. And in a very small numbers of mice, just like 2 mice here and just 1 mouse here, we see the same therapeutic effect. Okay, so that's the end. I know I'm at 40 minutes. We find that these gamma oscillations and their synchronization involving these parvalbumin interneurons seem to be really important for cognition. Targeting gamma synchrony might be a way to persistently rescue impairments in cognition. This might be done by these projections, these fibers that go from one side of the brain to the other side of the brain. Those might be like the key regulators here. And so now we're looking at this in Syngap mutant mice. We're also going to test the effects of clonazepam. We're going to target just these callosal projections, as I told you we just started doing. And we're also just going to look at these callosal projections that go from one side of the brain just to see, do they look normal? Is something wrong with them? That's what we're working on. So I really, again, want to thank the foundation, not only for funding this project, which is just started, like this is just the very beginning of it, but also, obviously for bringing this idea to me. And again, just thank all of our different funding sources. Thank the people who did this work. Kathleen was assisted by Carl Shi, who's a grad student at Columbia now. She was being assisted by Kira Wahlquist, who's a technician in my lab. We've had a lot of collaborators who've helped us set things up. And hopefully you have some questions now, but I can also be contacted later if you have more questions. Thank you all.
Wow, thank you so much for that tour de force. I learned so much today. It's really fascinating that that this small set of interneurons is so important for these sort of that switching task. And I have to say, for all, probably what all the parents on the call, Syngap parents on the call, are thinking is, yeah, my kid learns something and then it's really hard to unlearn it. It's really hard to have a change. And so I'm wondering what are you kind of most excited about next? And do you think that, yeah, what can you talk a little bit about where this would go as far as treatments? Like, I don't think we can do the,
Yeah,
would we ever be able to deliver gamma oscillations? Like, I think probably not, but the drug is really interesting. So can you just sort of go forward on that?
absolutely. Yeah. So, you know, obviously the reason we use this light stimulation, right, is because we want to do something that's very precisely defined. Because if you give a drug and it sort of works, but you don't know how it works, it's hard to make that better, right? It's hard to iterate on that. And so if we start off with something which we know exactly how it's working, then we can design our drug treatment or whatever to try to optimize that.
Well,
Now,
the light, and just to clarify, the light is associated with these channels, right? This is an engineered
that's
system.
right. This is a totally engineered approach, right? So
Yeah,
we are introducing these channels to make the neurons sensitive to light, and then we use the light to drive those neurons how we want.
yeah.
So, there's the most obvious thing is like, oh, maybe we could use clonazepam, right? This is a benzodiazepine. It's a drug which is used all the time for all kinds of things. It's, relatively safe. And maybe that's a way to boost gamma oscillations and their synchrony in the brain. That obviously worked in our DLX56 mutant mice. Now, it may or may not work, in other settings. And in particular, what we've found is if we are actively inhibiting these projections that go from one side of the brain to the other, then clonazepam doesn't work because presumably it's boosting those connections. And so it's sort of, those connections have to be like in a place where they're kind of working, but not working quite strong enough. And that may or may not be the case in say, Syngap mutant mice. But we're going to try that. We're going to try clonazepam because maybe just giving low doses of clonazepam is sufficient to boost this gamma activity that's going to be important. Other things we can do. The reason I spent so much time emphasizing this point, like, oh, we found these neurons that go from one side of the brain to the other side of the brain, and that seems really special, is because this is a really small group of cells, as you said, and that makes us think maybe those cells have special molecular targets. Maybe we could identify drugs which would bind to just those cells and modulate the activity of just those cells. That obviously requires like figuring out what genes those cells express and what kind of receptors they have. But that's all very doable. It'll take some time. So those are the immediate next steps, I think, is to kind of like, if in thinking about translation would be, how else can we boost gamma activity? How can we target these cells? What is the state of these cells in Syngap mutant mice, for example?
Can I jump in, Catherine, before you get into really more cool science questions?
Please.
I just want, well, Professor Sohal has this slide up. I just want to call out the top left, and like that list of funders is kind of like Sesame Street when you had those 4 boxes and one of these kids was not like the others, right? I think Sesame Street. I think Syngap Research Fund next to DARPA, Safari, McKnight, Brain Research Fund, and these NIH is, if you're a Syngap family or a Syngap donor, this is exactly why we do what we do, right? Like, this is what I talk about when I talk about catalytic capital. Like, we cannot afford to fund every single cool idea out there, but when we get a chance to get a lab like Professor Sohal's lab, who works with all of these different organizations and does this level of work to turn their attention to Syngap 1, which is, I think, definitely one of the up and coming monogenic developmental and epileptic encephalopathies, but it's just one. And I think it's everybody watching this should be really happy to know that this lab and this level of work is being done on our gene, with not only that, but with direct translational implications, right? Like, can clonazepam help us build on this insight or other PAMs, and maybe help us help our kids. So I just want to do a victory lap there. Point one. Point two, I want to give Dr. Shilpa Kadam a shout out. She's been an ally of Syngap One, and she's on our scientific advisory board, and she's just always doing us favors. And so I just want to thank her for connecting us to this. And then the third point, after I say thank you again for that presentation, was really excellent. It's really hard to share high science and make it accessible to mortals, and I think you just did that beautifully. But here's my question. When you talk about clonazepam, there's another researcher right now at Vanderbilt, and I've not, Professor Sohal, if you want to say, let me come back to you, that's fine, because I've not teed this up at all. But it's really interesting. He studies catatonia, and he's just put out a case study, and he's putting out a paper where basically he's saying he thinks catatonia is underdiagnosed in some of our kids. And he's saying one of the therapeutic implications is that instead of putting our kids on antipsychotics, he wants psychiatrists to think about maybe using benzodiazepines as a more effective strategy to treat them. Yeah. And when you talk about clonazepam, I'm just thinking out loud about that, because a lot of families are having this conversation with their psychs, right? And should we go down the Abilify risperidone pathway, or should we go down more Ativan or other benzos? And does this work? Does this work tie into that, or is this a bridge too far and I'm just anchoring on the clonazepam?
Yeah, no, I think it does tie in. Obviously, I don't want to get ahead of myself in giving specific treatment recommendations, because I'm a psychiatrist, and I can share my experiences, but obviously I'm not necessarily working with this patient population, and obviously each individual kid is going to be different. But if we think about catatonia, catatonia is something where, it seems like, people are stuck, and it seems like giving patients a sedative would be the thing you wouldn't want to do when someone is stuck. But actually, it has this amazing kind of, again, like almost magical effect where individuals will all of a sudden, start moving and talking. And, when you ask them and they're able to describe their subjective experience, which they can't always do super well, but they'll say something like, I was just stuck on this one thought and I couldn't stop thinking about it, and because of that I just felt like I had to, couldn't move, and somehow through a mechanism we don't fully understand, clonazepam and other benzodiazepines are able to get people unstuck. And so we don't know exactly what that mechanism is. It could be kind of a seizure-like effect, and maybe that's how the benzodiazepine is working, but it also could be something like what I'm saying, where there's normal brain oscillations that help you be flexible in your behavior and thinking, and that a dose of clonazepam helps to boost those. We don't know, but it's certainly a very plausible explanation. And so I do think it is plausible to think this relates to that idea, but again, there's much to be figured out.
Yeah. And now I'll give it back to Catherine. We also have, I think, one hand raised, but I will say that if you survey the BCBAs who work with a lot of different populations and work with Syngapians, there are a lot of people who would say Syngapians perseverate like insert your favorite term here, but I mean, the perseveration that you see in our patients is next level, yeah. Right? So that squares very much of what you just said.
Yeah.
Back to you, Catherine. Thank you again, Professor.
Yeah.
Thanks. Hey, Aaron, I saw you had your hand up for a moment. Is that, yeah?
Yeah. Yep. Thank you, Catherine. Yeah, to Mike's point, I mean, the perseverations with Jackson, who's 19 now, are quite off the charts in many different ways. But my question is more directed towards many of our patients are on clobazam, the Onfi, again, a benzo, but based on the molecular structure difference with clobazam, what is there a possibility that there is, based on the drug targeting, a benefit for many of those patients who are on Onfi clobazam?
So yeah, there's two or three different kind of things to think about here. I don't have like a definitive answer, but I can tell you how I'm thinking about this, right? Yeah. So, for the most part, we think about, the dose and duration as being the big differences between different benzodiazepines or benzodiazepine-like drugs, right? So some are more potent than others, some last longer than others, and mostly that's why we choose one rather than another. And for the most part, the doses that we use are doses that reduce anxiety and cause sedation. And maybe we have some doses that just reduce anxiety and don't cause as much sedation, but that's really narrow window there. What we had found, these pro-cognitive effects were at sub-anxiolytic doses. So we basically, tested our mice, found the dose at which, the smallest dose at which we could see any reduction in anxiety, and used half that dose. And that had like no sedative effects as far as we could tell. Now, so it's possible, first of all, that the doses that are being used of all these drugs are causing some sedation, which might counteract the pro-cognitive effects. I don't know. The second thing is that, even though we think about benzodiazepines, all these drugs, as just sort of enhancing inhibitory neurotransmission, it turns out that when GABA is released, there are lots of different receptors it can bind to, and different groups of cells tend to work through different receptors. So these like parvalbumin interneurons seem to target specific GABA receptors, and even the long-range projecting ones might have very specific ones that they target. And the reason I bring that up is because now drug companies, I say now, this has been going on for some time, I've been thinking about how to differentiate, like how to maybe tweak the molecular structure of benzodiazepines so they'll bind to some GABA receptors instead of others. This is why we have Ambien, right? This is essentially how Ambien works. It's a benzodiazepine-like drug that binds to some of these receptors better than others, which is why it produces more sedation and less anxiolysis. It's also why it doesn't cause muscle relaxation and paralysis, which is why sometimes people take Ambien and then they sleepwalk and like eat or even like leave their homes and try to drive. So that's sort of how to think about the question you asked, but I don't know the answer if clobazepam or clonazepam or different, like which of these drugs would be the best for what I'm talking about. I don't know. But anyway, the point is it could be different for different drugs. Yeah.
Thank you.
Yeah, thank you. We had a second question in the Q&A that is similar from our clinical director, Marta Tehia, MD. So she says, can you test if there's a difference between some of these different drugs? Is that part of the plan or not yet?
Yeah, I don't think that's a plan to test different drugs right now. I think, I think that, we definitely, our plan is to test clonazepam and see if that works. If it does, then I think, yes, then there would be a question about like, should we compare different drugs, or is that something that's better done in the context of like a pilot clinical study as opposed to like, it's at the point at which that information is more useful and to look at in humans than in mice. I think we would be looking to do the proof of concept and the mechanistic work in mice, right?
Right, yeah. Makes sense. Okay, and then we have a comment from Minnie Zhu. She says, I don't have a question. I just want to mention that your research is remarkable. Yeah, absolutely. She says, I'm always really interested in gamma oscillation because my daughter started the low-dose perampanel at age 2 from Dr. Kadam's research. It improved her gamma dysfunction from lack of Syngap. I can attest that she does not have perseverance in her behaviors and a lot more flexible and understanding and learning activities of daily living.
Yeah, that's amazing to hear, Minnie. Thank you for sharing that. I just have to say, yeah, it's really amazing to me to see, to hear, like, obviously I put up these figures and like, oh, we're amazed to see this thing, but it's way more amazing, obviously, if you see it, in your child kind of happening in front of your eyes in real time. And we haven't, I mean, whether or not to try perampanel is a good question, because it does sound like this is a medication which is widely used in this patient population. And so maybe that's something we should try in the mice to see what its effect is. That is something for us to think about.
I want to chime in there. The clobazam is widely used.
Okay.
Perampanel, Minnie's case is exciting, and I think she got two things right. She started young and she kept to low dose. The on-label dosing for perampanel, many of us took the Dr. Kadam paper to our doctors and got a script for perampanel and had a kid who was really like radically more cognitively capable for a week, and then had behaviors that were off the charts even for a Syngapian. And many, myself included, stopped. And I think
Yeah. Okay.
I then had to go back to doctor and say, give me low dose. Don't give me tablets, give me liquid.
Yeah.
And even then, the behaviors came up. So we have had a couple of success stories on perampanel in humans. We have had a lot of, my son included, kids who went really aggressive to the point where we had to stop and then realized we had a drug with a pretty significant half-life. So and I think there's definitely a there, right? It has an impact. The kids get so much smarter, but that then has this, I don't know what, where the behaviors are just insane. And so
Yeah.
I think both things are a thread to pull on, but
Yeah.
many of our kids are on fee.
Yeah.
A few of our kids are still on perampanel.
It's good to know. I mean, again, this speaks to why we, are also trying to just focus on these cells a little bit, as maybe we can use what we learn about the biology of these cells to find more specific ways of targeting them, because obviously all of these drugs, clonazepam, clobazam, perampanel, they go throughout the brain. As you say, they accumulate, the dose builds up, and so then you get all these effects on all kinds of things that lead to whatever you want to call it, side effects, adverse effects, emergent things. Yeah, that's interesting to know.
Yeah, and I just want to add on with behaviors, and there's a lot of different types of behaviors, but for Syngapians, it's often aggression and self-injury, and just really, really severe in both categories.
See, yeah.
Okay, let's see. It looks like we might have one more in the Q&A. Yeah, Melissa Smith says, for the mice tested with the rule change and had been treated with the channel rhodopsin repair and were repaired, were they then tested with a new novel rule change, and was the repair still there?
Yeah, so when we do a subsequent change in the rule, we're using different it's not like they're learning the same exact rule change. It's would be to a new rule that they haven't necessarily seen before. And so, but it's also not a completely different task. But I think part of the question is is this long-lasting effect we're seeing, them understanding this particular task and the particular things that they were shown before, or is it some plasticity happening in the brain that might improve their performance on lots of things? And it seems like a simple question to answer, but I would say actually it's in these DLX56 mutants, it was hard to find a lot of tasks that they were impaired on. These mice are sort of in this Goldilocks zone where they're impaired on this task, but not a lot of other tasks. And so this might be something we would look at more easily with the Syngap mice. But kind of a related underlying question there is, when we do the stimulation, are the mice just learning one thing better, or is something in their brain changing? And we're doing that experiment right now, where we're looking for long-term plasticity of brain pathways following this intervention. And we think we may be seeing evidence for that. Yeah.
So are you seeing, are you measuring the EEGs with these mice? Are they all inside the brain, or can you do anything?
Yeah, this is a good
outside the
question.
brain?
We do usually, there's a lot of, talked about a lot of different experiments. In the DLX56 mutant mice that I talked about at the beginning, we did measure EEGs from them. We always just implant the wires or skull screws inside the brain in mice. That's just the way we do EEG there. And we assume this is a comparable signal to what you see with EEG in humans. Not exactly the same, but maybe has some similar, to first order is the same. And I don't know if you're asking that because you're wondering what changes about their EEG, or what changes about, or if we're finding other things like seizures or.
Yeah. No, I was just wondering how applicable will this be to a human EEG? So I don't know enough about neuroscience to know with the PV interneurons
Yeah.
Is that something you can even see on a human EEG, or is this kind of
Yeah.
a
So
subclinical thing that isn't going to translate?
So the whole reason we're interested in gamma oscillations, right, is because these are originally described in human EEG, and you can take a low quality EEG, like one of these headband EEGs, and you'll see gamma activity. And, you see that, the EEG does not look the same on all parts of the brain. And when someone is doing a cognitive task that requires them to use cognitive control and executive function, you'll see more EEG activity in the front of the brain, so there's some localization. Now, the localization to brain region is obviously not going to be as good with scalp EEG as it is with, electrodes implanted in the brain. And obviously we can't say anything about cell types, right? So like we're using these genetically encoded voltage sensors, which are like fluorescent proteins, because we can target specific cell types. And it's kind of like measuring EEG from one point in the brain and from one cell type in the brain. Whereas EEG is kind of a spread out, it's like not perfectly localized, and it has zero specificity for one group of cells versus another group of cells. So, but ultimately the reason we're studying brain oscillations is because this is something which was discovered in EEG, and we think we should be able to take what we're finding back to humans and measure their EEGs and see something that we can associate with these very specific cellular changes. Yeah.
Okay, great. Well, I look forward to hearing all the progress reports in your work, and I can't wait to have another webinar when you have more Syngap findings.
Yeah, no, I look forward to it as well, because as I said, these conversations have been incredibly stimulating and really gotten us to think about things. And here too, I'll start thinking about these different drugs and how we should incorporate that into our work for sure.
Well, I don't have any more questions. Mike, do you?
No, I want to note that this is our second lab that we're working with at UCSF. We also have the pleasure of working with the Wilsey lab, and sometimes our patients stop by the Wilsey lab to do nasal nitrous oxide readings. So if your lab is ever interested in meeting, I'm always amazed when there's geniuses like your postdocs working on Syngap mice, developing this deep expertise in our gene, and they've never met a human.
Yeah.
So we do have humans who are seen at UCSF and who go to Helen's lab. So if you would like, Dr. Wilsey's lab, if you ever want to visit from a family, just let us know. I think it would be, sometimes I find that labs enjoy meeting the human.
Yeah, I think that could be great. Actually, some postdocs in the lab have asked specifically for this kind of thing. So I think that would be really wonderful. And so I'll follow up with you about that, Mike.
Great. Thank you again.
Yeah.
We're really excited about this.
All right. And again, people watching later, feel free to reach out to me via email if you have questions.
Yeah. So thank you so much, Dr. Sohal. What an incredible talk and amazing science. Really appreciate it.
Yep.
Okay. Bye.
Bye, everyone.